Hello, everyone, and welcome to part 3 of our 4-part series on 3D culture and analysis. How to analyze, characterize your 3D culture. I'm Christy Jewell of LabRoots, and I'll be moderating the session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. Now let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question and click Send. Questions will be answered following the presentation. If you experience technical problems seeing or hearing the presentation, just click on the Support tab found at the top right of the presentation window, or report your problem by typing it into the Ask a Question box located on the far left of your screen. This is an educational webinar and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right of your presentation window and follow the process of obtaining your credits. I now present today's speaker, Chris Langsdorf, Staff Scientist, Thermo Fisher Scientific. Chris's complete biography can be found in the Biography tab at the top of your screen. Chris, you may now begin your presentation.
All right, thank you, Christy, for the introduction, and thanks to you and LabRoots for hosting this webinar. And also thanks to everyone in the audience for joining us as we discuss our research on labeling and detection in 3D models, specifically with fluorescent reagents. So today we'll break the webinar into 3 sections. First, why are we interested in analyzing 3-dimensional models as a transition from 2-dimensional? Second, how we can visualize and analyze these 3D models, specifically what instruments are valuable for this. And then third, what are the key assays and reagents we use to label our cells to analyze different processes in these 3-dimensional cell models. So first, why are we interested in 3-dimensional models? So typically we'll do fluorescence microscopy on cells that exist in a monolayer on a flat plastic dish, but increasingly we're seeing researchers transition to a 3-dimensional spheroid model, where we know that growing cells in a monolayer, as we see here in the quote, that this is fundamentally dissimilar from how cancer cells exist in a 3-dimensional tumor. Things like the growth rates, morphologies, metabolism, and the interaction of cells with each other and with their substrates are very different in a 2-dimensional versus a 3-dimensional model. Now, if we look at this in more detail, as far as different considerations for these 2 different culture systems, we see that with 3D models, we expect a more natural cell morphology and a more representative expression patterns of proteins. Also critically, the diffusion patterns of drugs, growth factors, things in our culture medium to our cells vary across cells in a 3-dimensional system. On the potential downside, there are things we need to optimize. So our experimental workflows, the assays that we're used to using, need some special tips and tricks to use these appropriately in a 3-dimensional system. And also, we're now dealing with an added level of complexity that can add some technical challenges. So today we'll look at how we can overcome these challenges and apply some of our characteristic assays to these 3-dimensional models. We expect in a 3-dimensional model to see a more representative morphology that better relates to an in vivo tumor, where we can see the reproduction of characteristic differences in cellular activity. On the left here, we see a difference in proliferative ring. So we can see in green, as illuminated by our cell event caspase 3,7 green reagent, we're able to visualize the apoptotic or necrotic core, those cells that are isolated from growth factors and oxygen and become necrotic. On the right, we also illustrate with our image hypoxia green reagent that the core of these three-dimensional structures is present in a reduced oxygen tension. And so, as we'd expect, there's a gradient of not only oxygen, but also metabolites and nutrients across this three-dimensional structure. So this has implications for not only how we label our cells, but also what biology is occurring in cells depending on where they exist throughout the structure. Now, when it comes to actually imaging through this three-dimensional structure, we can use wide field imaging in which the in-focus image from our plane of cells of interest is mixed with the out-of-focus light from regions outside the focal plane. And so we see essentially a blurred image here. However, with laser scanning confocal microscopy, it's possible to image just a thin slice out of this thick specimen and reject light from other planes that are not in our desired focal plane. So we have better resolution of cells deep within this three-dimensional structure. And so we'll look in more detail at instruments that provide this capability of imaging into larger structures. And so here's an example using actual fluorescence where we can visualize live and dead cells both with wide field and confocal imaging. And it's clear that the cells on the interior of this three-dimensional structure are visible at greater resolution when we're using confocal imaging. So today we'll look at not just the instruments that we use to image and analyze three-dimensional cells, but also how we can use a range of different fluorescent reagents and fluorescence-based assays to label these structures to provide insight into the biology we're studying. And so first we'll look at the instruments that we can use to visualize and analyze these three-dimensional models and how we can use these in optimal experiments to characterize our 3D structures. So first, when it's time to form our three-dimensional spheroid in this case, it's critical that we have control over the number of cells that are being used to grow a particular spheroid. So using an instrument called the Invitrogen Countess 2 automated cell counter, we can very quickly, using 10 microliters of our cells, obtain an accurate measurement of concentration and viability so that we can seed the correct number of cells and a consistent number of cells per well in our 96-well plate to grow spheroids of a consistent and controlled size. Now, when it's time to grow our spheroids, we recommend a plate called the Nucleon Sphera 96-well plate to grow consistent spheroids with a homogeneous spherical shape. And so as we see here on the bottom right, as we increase the number of cells per well, we see a larger spheroid structure as we'd expect. Now, using a similar plate that's not treated as the Nucleon Sphera is, we see occasionally non-ideal formation of spheroids. So two things that are critical, counting our cells and then using an appropriate plasticware to grow a nice round spheroid. And so now when it's time to image these and evaluate the biology occurring inside these three-dimensional structures, fluorescence is really ideal because it provides us a way to not only label our cells and distribute a dye throughout the structure to report different functions, but to detect light given off by that sensor. And so with fluorescence, we're able to maintain the three-dimensional structure and the interactions between cells in this structure. And using different probes, we can measure the abundance or also the localization within the structure of different proteins or different processes or functions that are occurring within the spheroid. So we'll look at several examples of those when it's time to look at assays and reagents. So for visualizing and analyzing our 3D models, we'll look at three different instruments. where, as we see in the first quote here, visualization is fundamental to document the differentiation on a daily basis. And our second quote, that this can be a time-consuming process to grow a three-dimensional structure. We need a robust imaging system to be sure that things are going correctly during this process. So we'll look at three things: the eVOS imaging systems, our high-content analysis platforms, and then also the VarioScan line of plate readers for whole well analysis. So first, the eVOS M7000 instrument is a wide field microscope that's fully automated with high-speed acquisition, so that you can scan multiple wells, 96 wells if you like, but also with the power to create Z-stacks or image a wide area and tile or stitch those images into one large reconstructed image. It's also compatible with an onstage incubator, so you can control not only the temperature, but gases and humidity on your cells to perform time-lapse live cell imaging. And finally, once we've acquired our images, we can do a 3D image analysis with the Invitrogen Celeste image analysis software. And so one example of how we use Celeste is to essentially create a mask around our entire spheroid. And we see here in red, the red circle, we've identified our spheroid, and the yellow circle, we've identified our background for one spheroid. And we can apply this mask to every image in our experiment and quantify the fluorescence in our different markers that we've applied. And so in this case, we see a 96 well plate full of, in this case, A549 lung cancer spheroids, where we can image every single one of those and not only have the qualitative information of the image, but we can extract quantitative information. So first, on the top right, we can look at the size of the spheroid, and we see after a drug treatment that the size, the average size or average area of every spheroid has decreased after treatment. And also in the bottom panel, we see our treatment increases the level of a green signal reporting caspase 3 and 7 activity. And so we see our treatment significantly increases the signal of caspase reporting apoptosis. So we can perform this kind of analysis on the eVOS microscope. Another system that's valuable for this kind of high content analysis is the Insight CX7 laser high content analysis platform. And so from this, we can provide the same image-based quantitative cellular data. So the CX7 laser has a 7-channel laser-based illumination, also with a powerful HCS Studio software to analyze your images. And so we can collect data on a per cell basis and multiplex with several different markers. This instrument is also live cell compatible, and so we can use the same eVOS onstage incubator to control the environment inside the instrument to keep our cells happy and growing for kinetic experiments or longer term experiments. Now with high content analysis, we have a slightly more sophisticated software which provides an automated acquisition and analysis that helps us improve and speed up our data capture and our downstream analysis. So essentially what we do is label our cells with something, in this case label our entire spheroid with the dye like Hookst 33342. This will provide a landmark on each individual cell so that we can distinguish and put a software-based mask on each individual cell. Then we can apply a second or a third probe. In this case, we'll use a TMRM, and we see the image in this channel. And we'll ask the software to create a mask identifying not only each cell based on their nuclear stain, but also to identify the amount or the brightness of signal in our second channel. And so by doing so, we'll provide a cell-by-cell measurement of our mitochondrial membrane potential in this very large three-dimensional structure. Now that we have this information, we can plot it not as an image, but as a fluorescence-based histogram, and see the average, or the mean intensity on a per-cell basis of this probe. And so now we can extrapolate this and look at our entire spheroid and get an average intensity of, in this case, mitotracker orange or cell tracker green across the entire spheroid. So a cell-by-cell quantitative analysis extracted from a single image. So in addition to the quantitative analysis, we can perform stitching on this instrument as well. So if we have a larger structure to image, we can take a large number of different images, in this case with 4 different colors, and merge not only the 4 different colors together, but merge all of the single images together to provide a very large and high resolution image of this large structure that we want to image. So moving on to a third way to analyze our 3D structures, here we'll look at a microplate reader called the VarioScan Lux. This is a multi-mode plate reader that is very versatile, giving us up to 5 different types of measurements. This could be absorbance, this could be fluorescence, luminescence, time resolved FRET, and these can be run as both endpoint or kinetic assays. This also has a powerful but very easy to use ScanIt software for powerful and intuitive analysis. And also, we've tried to automate a lot of the experiments using an online protocols library with about 130 different pre-made protocols. So let me show you what the data will actually look like from this instrument and how it might relate to an actual microscopy-based instrument, where here with the microplate reader, we're detecting all of the light from an individual well of a 96-well plate. So we see here on the left several dose response curves of A549 cell spheroids that were treated with different doses of cambogic acid, and we're evaluating the signal from every well of cells at different time points. And we can compare this to the same plate, the same 96-well plate of spheroids as analyzed by our high content analysis system. And so with high content analysis, we have the images to go along with every one of these data points, but it takes a bit longer to acquire those images. If we just want quantitative information, the VarioScan plate reader can provide similar results, but in a much quicker analysis. But we can report similar toxicity profiles with both instruments. It really just depends on what the desired output is of this experiment. Is it just the quantitative evaluation of viability, or is it that plus the localization of individual cells within the images? And so that was 3 different ways we can look at imaging and analyzing spheroids or other 3-dimensional structures. Now let's move on and look at the different assays and reagents, fluorescent dyes that we'll use to label our spheroids, to label our 3D structures. And also we'll pay attention to some tricks that we've found improve the ability of some of our conventional dyes and reagents to perform in these more complicated models. So we need to do a few things. We need to show that we can adapt existing reagents to these 3D models and show how a wide range of different methods can be used here. So we'll look at a few different things. First, at functional readouts and how we can measure different things like viability, proliferation. We can look at cell health as evidenced by things like apoptosis, reactive oxygen species, and also in this category, I include things like immuno-oncology assays, and we'll show several examples of those and how we can incorporate immuno-oncology assays with primary immune cells into these models. And third, we'll look at how antibodies are used here, and including antibody-drug conjugates. So when we're looking at spheroids, a common model is to grow spheroids out of cancer cell lines. And a common readout here is how can we induce cell death pathways. And so a common readout is viability, where here we're showing the Invitrogen live dead cell imaging kit applied to HeLa cells that were grown into spheroids on our Nucleon sphere plates and treated with different doses of niclosamide. So we see our cells going from green and healthy to red as cell death is initiated, and we see a dose-dependent response here. So this is one common assay, but as I mentioned, there are many other things that we might be interested in measuring, things that are potentially measuring pre-lethal toxicity as well, things like mitochondrial health, reactive oxygen species, apoptosis, or proliferation and cell cycle. So we'll look at examples of some of these other assays now. So first, let's look at a measurement of caspase activity, both a qualitative measurement from an image of spheroids and the related quantitation on a single cell basis. And so we can see here a dose response of cells. These are again A549 spheroids treated with increasing concentrations of a drug, niclosamide. And we see very little green signal in our control, but as we increase the drug concentration, we see individual cells turning very brightly green fluorescent. And so we can do two things. Based on just our white light image, we can plot the size of this spheroid and show that increasing drug concentrations shrink the spheroid. They lead to death and shrinkage of the spheroid. But also we can correlate that with killing as reported by our cell event caspase 3.7 probe and show quantitative increase in signal as our drug concentration is increased. So this is somewhat a late measurement of toxicity. A somewhat earlier measurement of toxicity is provided by reactive oxygen species detection. And so we can use a probe such as the Invitrogen CellRox Deep Red reagent. We'll load our entire spheroid or plate full of spheroids with this reagent. And as we treat with different concentrations of a drug, menadione, we see an absence of red signal in our control cells, but we see a significant increase in red signal reporting the presence of reactive oxygen species as we increase the concentration of our drug. So we can see this in the image, but we can also quantify this and illustrate the increasing, significantly increasing presence of reactive oxygen species as we treat with this drug. And so a tip here to perform this experiment that actually is common in 3D models is that we may need longer incubation times to achieve optimal signal. And so here we needed to increase the incubation time for both the CellRox dye labeling to allow time for it to penetrate through this larger structure. But we also needed a longer incubation time for the drug for the same reason, to give it time to penetrate into the thicker structure. And so this is, as you're translating your experiments to 3D, this is something to watch out for with a lot of different assays and systems to increase the incubation time, and in some cases increase the concentration of your reagent or your drug. So let's move on and look at another example, this time at cell proliferation in spheroids. Here we're using two different probes. One is Invitrogen's ClickIt-EDU. And so EDU is an analog that's incorporated into DNA of replicating cells during S phase of the cell cycle. And we can detect that here in individual cells colored green. Now, Ki67 is a marker expressed in proliferating cells as well in the nucleus of proliferating cells. Here we've pseudo-colored that red based on an antibody-based detection. And so we can see individual cells within this top spheroid that are either green or magenta, reporting a positive signal for either or both of those probes. In the bottom spheroid in this panel, we've treated with hydroxyurea that inhibits proliferation, and we see an absence of green signal from our EDU, and we see a minimization of our Ki67, or I should say a reduction in our Ki67. And so when we actually quantify this on a single cell basis, we see in our two bar graphs that the Alexaflor-488 EDU signal is eliminated by treatment with the hydroxyurea. We see an absence of signal there. With the Ki67, we see a slight reduction. And also the bar graph on the right, we can plot the actual area of that spheroid and show that treatment with hydroxyurea leads to a slight decrease in the cross-sectional area of that spheroid. So this is a way to multiplex two different markers of proliferation, as well as to look at just the size of our spheroid. So I mentioned that we're not just transitioning individual functional assays like this, but also multi-cell immune-based assays. So let's look at some different assays and reagents for immuno-oncology and how we translate these into three-dimensional models. So here we're looking at a CAR T killing study of some colon cancer spheroids. And so we can see three different spheroids that were formed and then treated with CAR T cells designed to target them at different effector to target ratios. So on the left, we have no T cells. On the right, we have lots of T cells. And in the middle, we have a middle amount of T cells. And so these spheroids were fixed and permed and labeled with antibodies against cytokeratin, reporting our tumor cells, and CD3 epsilon, reporting our CAR T cells, and of course, hooks for our nuclear counterstain. And we can see as we increase the number of T cells, we see this clear degradation of the three-dimensional structure of our spheroid as the cells as the spheroid cells are killed. So we're not actually reporting killing by this assay, but we can by adding cytotoxicity markers such as our cell event caspase 3-7 reagent or dyes like image dead green. And so let me show you another example and how we can, instead of doing a fixed analysis like this, we can actually do a live analysis of immune cell killing in 3D. So let's look at a natural killer cell antibody-dependent cellular cytotoxicity assay. So we'll grow spheroids in our Nucleon Sphera 96 well plate from SKBR3 HER2-positive breast cancer cells. And then we'll label them here with Nuc Blue Live, which labels the nuclei of live cells with blue. Then we'll add cell event caspase 3-7 reagent. We see a few individual spots of green reporting cell death. But there's not actually killing happening here because we have not added our natural killer cells. So now let's add to this experiment in a separate well with a separate spheroid. We'll add some primary human natural killer cells. In this case, they were labeled with the dye cell tracker deep red, so we can identify them over time. And what we see here, after 4 hours of incubation and analysis with our Cell Insight CX7 laser high content analysis system, is we see that our natural killer cells penetrate to some extent into the spheroid, and we see individual spots of green reporting killing of individual cells. But we've not added trastuzumab, so we could say this is antibody-independent killing. Look at antibody-dependent cytotoxicity. Let's add trastuzumab or Herceptin to a third well in this plate. And now we see not only more robust penetration of these red NK cells into the structure, and more individual green cells reporting cell death, but the entire structure is degraded. We're actually seeing this entire tumor spheroid falling apart as the level of toxicity is so great, the level of killing by our NK cells. And so again, this is a live cell model where we're imaging live after 4 hours. We can do something similar with live T cells, or another currently relevant model for cancer therapeutics is with antibody drug conjugates. And so we can use the same antibody, say Herceptin, and use a kit called the Invitrogen Site-Click antibody labeling kit that allows us to site-specifically label antibodies with either a dye or a drug on a conserved region of the antibody FC domain. So essentially, we'll follow the protocol of this kit to site-specifically attach either a dye or a drug to this carbohydrate domain on our antibody. Now, when we put this on our tumor spheroid with cells expressing HER2, we actually created 2 versions of this, one with just a dye and one with a dye and a tubulin destabilizing drug, monomethyl auristatin E. And so if we are to image these cells, we see the Herceptin labeled with just the dye, and the dye in this case is Estivo-Froto red. We apply this to a spheroid overnight, in this case 24 hours, and we see that this antibody has been taken up and actually internalized to the lysosomes of every red cell in this spheroid. And we see a couple of green spots reporting spontaneous cell death. But if we take the same antibody and attach both the dye, the Estivo-Froto red, and our monomethyl auristatin E, now we see not only red signal in individual cells from internalization of this antibody drug conjugate, but we see bright green circles reporting death of individual cells as this toxin is internalized and kills specifically individual cells. So several different models we can use depending on what immuno-oncology modality we're interested in. So one final thing to look at as far as general advice for spheroid models is when we're imaging these larger structures, we need to be aware of photobleaching. And this is the irreversible destruction of a fluorophore as it's exposed to excess light. Now we can prevent this using anti-fade reagents. A reagent that fits this bill that has existed for several years is Invitrogen Prolong Diamond that provides very valuable anti-fade protection in thin specimens, but for thicker specimens such as spheroids or these cortical neuron organoids, we need something new called the Invitrogen Prolong Glass. Prolong Glass has a refractive index that's higher and better matched to that of glass, our glass cover slips. And so we see less light refraction. It gives us better penetration of our light in the Z direction, allowing us to better visualize cells on the interior of these larger structures. So this prolonged glass is very valuable for this type of imaging. A couple of other general guidelines. First, with respect to labeling and labeling with fluorescent reagents, we need to optimize our labeling conditions. So we looked at several examples here today where we have optimized many common reagents for use in 3D models. If you're embarking on a new protocol or transitioning your current methods from 2D to 3D, I'd look at optimizing those as far as time and concentration, and really seeing what works best for your system. And things we're looking out for are heterogeneity in labeling. Potentially that could be higher signal on the exterior of our structures as our dyes don't penetrate, or we can see increased backgrounds from these thicker structures. So things that are valuable here are optical sectioning using confocal microscopy, as we saw with the CX7 laser instrument, where we're able to visualize cells in a single plane of a single optical slice through our specimen. And we need to watch out for phototoxicity, especially if we're doing analysis of live spheroids, where excessive exposure to light can be toxic to those cells. To summarize, we looked at three different categories of three-dimensional analysis. First, why we would want to transition to a three-dimensional model. Then we looked at the instruments that allow us to perform this type of analysis, and then several different examples of assays and reagents that we find have great value for studying biology in this more relevant context. So thank you for joining us today, and I'll hand it over to Christy.
Thank you, Chris, for your informative presentation. A quick reminder to our audience on how to submit questions. Simply type them into the drop-down box on the left side of your screen labeled Ask a Question. Type your question into that box and click Send. Our speaker will answer as many questions as time permits. Now, any questions that we are not able to address in our live presentation here, or any of those submitted during the on-demand period, will be answered via the email address you provided at your time of registration. Okay, so Chris, your first question is, how can I reduce photobleaching or phototoxicity when imaging thick samples?
Thanks, Christy. That's a great question. And so, imaging these thick structures, we do have to worry about photobleaching of our fluorophores and also the toxicity induced in our cells by exposure to excess light. So for photobleaching, we can use confocal imaging and limit our detection to a single plane of light. We can also use the mounting media that I showed, specifically prolonged glass, that will preserve our fixed samples and prevent photobleaching or destruction of fluorophores in those models. So, thank you.
Thank you, Chris. Sorry, thank you, Chris. Our next question, how can I translate my current experiment into a 3D model?
Yeah, so good question. I think the points towards the end there was just general advice apply here, where really we may need to experiment a little bit as you're transitioning to a three-dimensional model. I'd highly recommend the Nucleon Sphera plates as a way to get started making spheroids with your adherent cell line of choice. And I think just allow some room for experimentation and optimization, where on our website on thermofisher.com, there's information available on how we've optimized several of the most common assays for 3D experiments. And so I think that's a good place to start.
Thanks, Chris. Now, let's go with this question. How did you make the round spheroids in this presentation?
Oh, yeah. So towards the beginning, we looked at a couple of different models. One where we're just using a standard round bottom 96 well plate. And we see that when we seed adherent cells into that type of dish, the untreated round bottom dish, sometimes we'll see multiple spheroids form. We'll see multiple larger structures form, but it's not just one round spheroid. And so we've really found in our lab that using these treated Nunclon Sphera plates, essentially we just aliquot our cells into every well and incubate overnight, and we have the nice round spheroids that are consistent and regular balls of cells for our models. So definitely recommend looking into those.
Thank you, Chris. Now, Chris, it looks like we have time for one more question, and I do see that we have more questions in our queue. So quick reminder to our audience that those questions submitted right now and any questions that do come in during the on-demand period, these will be answered via email. Okay, Chris, our last question. Are there any tricks to handling spheroids?
Yeah, thanks, Christy. Another good question where all of the different assays and reagents and dyes that we've discussed, these are things that we'll need to add to our cells, and in many cases we'll need to wash our cells, or in some cases fix and perm. These are all things that require media changes and manipulating our cells. And so if you're using a two-dimensional model of adherent cells, that's very easy because your cells are stuck to the bottom of your dish, and so you can be fairly vigorous with your washing and changing of fluids. With these spheroids, they're all just gently sitting in the bottom of this dish. They're not attached to anything, so we need to be a lot more gentle with our pipetting and with our media changes. Or as you see here on the final slide, we actually ended up with 2 spheroids in one well because of a more aggressive washing. And so just take your time, slow down on the pipetting, and gently aspirate your media, and very gently replace with your reagent of interest to make sure your spheroids not only remain in the well, but they remain as a nice intact ball of cells for your research. So thanks for the question.
Yes, and Chris, thank you again for your presentation on analyzing and characterizing your 3D culture. Do you have any final comments you'd like to leave our audience with?
Yeah, thanks, Christy. I think just to refer everyone to the thermofisher.com website, where you can find a lot more information on the protocols we've developed for this type of assay. I'd also like to invite everyone to join us for the next presentation in this series on 3D biology. So, thank you.
Thank you again, Chris. I'd also like to thank our audience for joining us today and for their interesting questions. And I'd like to thank our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. You will now be redirected to register for Part 4 on 3D culture and analysis. Hope to see you then. Goodbye.